We have asked Doug Turnbull, the director of the Core Genomics Facility at the University of Oregon, and several students to briefly discuss their research in our lab.
I'll hand you a page and we'll do it again.
I'm Doug Turnbull, that's D-O-U-G-T-U-R-N-B-U-L-L. I'm director of the Genomics and Cell Characterization Core Facility at University of Oregon. Yeah, so for Bill and Chris's research, they want to quantify the expression of thousands of different genes over time, and the assay they're using to do that is quantitative real-time PCR. So in order to set up those experiments, you need to do this in a multi-well plate where you have 96 wells on a little piece of plastic, and a very precise, consistent volume of several different reagents needs to be added to each well in this plate. If you have too much of one reagent in well A versus not enough of that reagent in well B, you'll get different readings that are actually wrong. So we need to set up these assays in a very consistent way, and fortunately here in the core facility we have a liquid handling robot that allows us to do that. It's a lot more reliable. It's slightly faster than a human doing it, but really the great thing about it is its consistency. It's the same volume delivered to each well in the plate every time.
So my name is Rudy Borozek. I am a doctorate student in the Bradshaw-Holzapfel Lab.
My name is Mary Wood. I'm a master's student in the lab.
So Mary and I are interested in the fitness consequences of the evolutionary transition of from biting to not biting in the pitcher plant mosquito, Wyomii smithii.
Specifically, we're interested in seeing how this transition has affected life history traits in our mosquito, such as pre-adult development, adult longevity, reproductive effort, and reproductive allocation.
Yeah, at the same time we've been imposing selection for biting for almost 20 generations now, and we hope to use this biting line to better understand the relationship between propensity to bite and disease transmission. So this mosquito is really unique, and the whole system is really unique, in that in the southern portions of its range, the mosquito will bite. Females of southern populations will bite. However, in the northern portions of its range, the mosquito absolutely will not bite, so it's totally lost the ability to do so. And this allows us to make really unique and important comparisons between biters and non-biters within the same species in their natural environment.
There are a couple different types of comparisons that you can make. One type, and what Rudy and I are interested in, is life historical comparisons, so seeing how traits like reproductive effort vary between biters and non-biters. But you can also make comparisons at the level of the genes, seeing how gene expression varies between biting and non-biting mosquitoes. And so this would allow us to determine the gene or genes responsible for the biting phenotype, and that isn't known currently and would make a really big impact in medical research.
So Nicholas Allen DePatty, Nicholas Alan DEPATIE, and my title is Master's Student in the Bradshaw-Holzapfel Lab. I'm interested in determining variation, if any, in the key genes that underlie the daily biological timing mechanism, which is known as circadian rhythm, in Wyomii smithii mosquito, and I'm doing this over a geographical range. So what I'm doing is measuring actual gene expression of the 5 core daily circadian genes that are involved in a transcription translation feedback loop. So what that means is we're studying elevational gradients as well as latitudinal gradients. So our latitudinal gradient is we have populations that range from the Gulf Coast of Florida all the way up the coast into Newfoundland, Canada, and our elevational gradients that we have are from sea level all the way up into the Appalachian Highlands in North Carolina. So we're doing this by using sensitive genomic tools called quantitative PCR, which allows us to measure gene expression levels at any sample we put in the machine to do these analyses.
My name is Nicole Kingsley. That's spelled NicoleKingsley. I'm the senior undergraduate researcher in the lab. My research concerns the degree to which dominance and epistasis, that is gene-gene interaction, has contributed to the evolutionary transition from a blood-feeding to a non-blood feeding lifestyle in Wyomii smithii. I'm also involved in the day-to-day maintenance and management of the lab, including the long-term development of the selected biting line.
Jack Benda, Jackbenda. I'm part of the bioinformatics team that is involved in gene discovery. So for example, we take the unknown contigs and singletons from the Waimea transcriptome and we compare them against genes in the NCBI database to find the unknown genes' names and functions. So right now I am working on the computer and writing script to simplify data analysis.
I'm Lauren Gomat. I'm LAURENGOEMAAT. I'm an undergraduate assistant here in the lab. I'm planning to get involved in an independent research project once the academic year begins. We currently have 200,000 Waimea smithii here in the lab that we are synchronizing in diapause to be used in future experiments. In these experiments, we replicate the natural environment by using the leaves of the pitcher plant. I'm grooming these leaves in order to keep the plants healthy and producing new leaves.
I am Liesl Benda, Lieslbenda, and I'm a research student at the lab. Key experiments in the lab are run in real pitcher plant leaves in computer-controlled environment rooms, which are programmed to replicate any environment from the tropics to the polar regions of Earth. I'm preparing insect meals, which are the exact biomass or weight that the pitcher plant leaves would capture in the field.
Leandra, Leandra Filiacci, Filiaci, undergraduate researcher. So we work together to care for the animals while learning experimental skills that can lead to independent research.
My name is Eric Jung, Erikjung. So my experiments involve how Waimea smithii uses the length of day to regulate its seasonal development from Florida to Newfoundland, as well as from sea level to mountain highland, North Carolina.
Hi, I'm Danielle McCann, Daniell McCann. Many plants are considered carnivorous because they're able to trap and digest their prey. The assumption is that carnivory for carnivorous plants is beneficial. No one has tested this assumption under controlled conditions throughout a year-long seasonal environment. My question asks if the level of predation by carnivorous plants is beneficial for vegetative and reproductive success of the host plant. This is important because many carnivorous plants are considered threatened or endangered in many places. The experimental determination of the relative importance of carnivory for carnivorous plants, as opposed to photosynthesis alone, which all plants do, is important for management practices for conservation of these unique plants.
Hi, my name is Daniela Lewis, DANIELIA Synchronous development enhances mating opportunities and avoidance of predation. Synchronous spring development enables us the application of control measures that maximize their effect on mosquitoes while minimizing the effect on the environment. My research concerns the interplay between the environment and mechanisms by which different mosquito populations become synchronous in their spring development. And the goal of my research is to be able to predict an optimal time to employ biological or chemical control measures that minimize the use on the environment.
My name is Ethan Kirsch, ETHANKIRSCH. Mosquito bloodborne diseases are responsible for over 500,000 deaths annually just in sub-Saharan Africa, and most of these occur in children. The ultimate goal of my research is aimed at eliminating biting in mosquitoes by increasing the costs associated with blood feeding or reducing the benefits. The important underlying logic here is that if a mosquito doesn't bite, it is unable to transmit these harmful disease pathogens. I'm currently looking at costs associated with blood feeding, specifically DNA damaging by heme or the iron associated with heme contained within blood. If we're able to manipulate the mechanisms that mosquitoes use to detoxify the blood that they ingest, we may be able to decrease the benefits of taking a blood meal and enhance selection on alternative forms of adult nutrition.
My name is Olivia Zito, that's OLIVIASIDOW. My responsibilities include the oversight and organization of lab activities. My work includes the vital training of students in the care for and rearing of vigorous stock populations used by everyone in their research, and in the day-to-day maintenance and coordination of the lab in general. My job is to see that the lab functions both efficiently and harmoniously so that students can learn how to do research, get excited about their own research, and also share that experience with others. In this lab, I have learned essential skills for the functional, financial, and personnel management that make for a productive, diverse, inclusive, and collegial workplace environment.
Hello, my name is Emma Rasmussen, EMMARASMUSSEN. Mosquitoes are known as the most deadly animals on Earth because they transmit heinous diseases such as malaria, dengue, and yellow fever. Biting and sucking blood is hazardous to mosquitoes because, among other substances, blood contains toxic amounts of iron. In addition to blood, mosquitoes also feed on carbohydrates and are attracted to sugar-baited traps. I will be testing whether iron tolerance varies between blood-feeding and obligate non-biting populations of mosquitoes. In addition to blood, mosquitoes also feed on carbohydrates and are attracted to sugar-baited traps. Iron could be added to sugar bait, along with other substances that specifically interfere with iron tolerance in mosquitoes. Hence, my research will contribute to the control of mosquito-borne disease using measures benign to the environment.
My name is Kasha Khalsa, spelled Kashakhalsa, and I'm a new undergraduate student with a particular interest in physiology. After becoming adept at raising mosquitoes, preparing food, and maintaining plants, I plan to work on a project aimed at the eradication of mosquito blood-borne disease.
My name is Travis Newman, TravisNeumann. I'm looking at 30 known genes in the mosquito Wyomia smithii, important in the lipid metabolic pathway. I'm looking at the genes particularly from the static hibernating state to the reinitiation of active development. The reason for looking at these particular genes is in hopes of finding a point in the metabolic pathway that could be interrupted in hopes of controlling mosquitoes.
My name is Mitro Rezonico, MITCHELLREZZONICO. In mosquitoes, blood taken from biting is assumed to yolk the eggs, that is for embryonic development. At the same time, carbohydrates are necessary to sustain adult development. Wyomia smithii populations in the north never take blood meals, yet still produce batches of fertile eggs. My research is aimed at determining which nutrients are both necessary and sufficient to sustain adult development. The understanding of these nutrients and the transformation of blood-feeding mosquitoes to non-biting mosquitoes is important in helping to understand how to eradicate blood-borne diseases such as Zika, malaria, West Nile, and dengue.